Picture this. You're in the most hostile, dangerous environment imaginable for human physiology. It's a place where intense cosmic radiation is actively breaking down your DNA. Extreme temperature swings threaten to destroy basic chemical bonds, and the complete absence of gravity is aggressively degrading your muscles and bones. Sounds like a total nightmare, right? Well, here's the wild paradox we're diving in today. This exact same extreme environment, deep space, might actually be the ultimate golden laboratory for discovering our next major pharmaceutical breakthroughs. Welcome to this explainer, where we're going to explore how the harshness of deep space could fundamentally transform the way we design and develop medicines. Okay, let's take a quick look at our roadmap for today. We'll start with the deep space pharmacy, then cover why space is different, microgravity and drug design, cosmic radiation and AI, formulations for long missions, and finally, limitations and Earth translation. All right, part one, the deep space pharmacy. To really understand why scientists are looking to the stars for medicine, we have to look at two massive colliding problems. First off, humanity is planning some incredibly long-duration missions. We're talking multi-year trips to Mars. But standard terrestrial pharmaceuticals simply degrade under cosmic radiation. They have strict shelf life constraints, and they just won't survive the journey. Second, back here on Earth, the global pharmaceutical sector is hitting a wall. Developing a single approved drug now costs an average of $2.6 billion. It's a massive bottleneck, and we desperately need transformative solutions to break through it. Before we get too far, let's just make sure we're perfectly aligned on our terms. Today, we are laser-focused on the early stages of making medicines, drug design and drug development. Drug design is all about identifying and building the precise molecular structures that target a specific disease. Drug development is the rigorous testing, modeling, and optimizing of those molecules. What we are not talking about today is mass manufacturing. We aren't looking at building giant, generic pill factories in orbit. Instead, we're talking about using space to invent entirely new, life-saving molecules before they ever reach a production line. Moving on to part two, why space is different. So, what is it exactly about the void outside our atmosphere that makes it such a powerful catalyst for biomedical research? Well, it basically boils down to four extreme factors that make up our extraterrestrial laboratory. First, we have microgravity, which fundamentally alters how cells and proteins grow. Second, there's deep space radiation, which induces unique biological effects and accelerates mutations. Third, we're dealing with an ultravacuum and extreme ultra-low temperatures that change how molecules behave. And finally, special resources, things like unique metabolic microorganisms and elements you just don't easily find on Earth. Let's break down the most impactful of these, starting with microgravity. Section 3, Microgravity and Drug Design. Taking Earth's gravity out of the equation doesn't just make things float around. It actually changes biology and chemistry at a fundamental structural level. To really appreciate this, you need to understand something called structure-based drug design, or SBDD for short. Think of a disease-causing protein like an incredibly complex microscopic lock. To build a drug that stops it, the key, essentially, you need to know the exact three-dimensional atomic structure of that lock. To figure that out, scientists grow crystals of these proteins. But here's the catch. On Earth, gravity causes convection and sedimentation in the liquid. The molecules settle unevenly, they bump into each other, and the resulting crystals often grow distorted and with pretty low resolution. But when you move this process to microgravity, it is an absolute game changer. The near absence of gravity completely eliminates that convection and sedimentation. The molecules can arrange themselves in a highly orderly fashion without being crushed under their own weight. The result? We get larger, beautifully ordered crystals with true atomic level accuracy. And this isn't just theoretical. Experiments aboard the International Space Station have already grown vastly superior crystals of HSB90, which is a protein linked to cancer, and beta amyloid, a key protein in Alzheimer's disease. They even achieved incredible uniform particle distribution for the cancer drug pembrolizumab. Space provides a high-definition blueprint that Earth simply cannot match. Now, what's really fascinating here is how microgravity impacts living cells. Spaceflight essentially puts human biology on fast forward. Astronauts can experience an average bone loss of 0.5% to 2% per month, which perfectly simulates severe osteoporosis alongside rapid muscle atrophy, known as sarcopenia. By cultivating human tissue up on the space station, scientists can trigger these metabolic imbalances in mere days. This creates an incredibly accelerated platform to test regenerative drugs like IGF-1. It lets us validate therapeutic targets for muscle and bone conditions way faster than we ever could using standard models back on Earth.
Okay, let's head into part 4, cosmic radiation and AI. We're shifting focus now to arguably the most dangerous element of deep space, galactic cosmic rays and solar energetic particles.
Outside Earth's protective magnetosphere, radiation levels are intense. And this radiation has a really fascinating dual nature. Yes, it degrades tissue and induces severe DNA damage, which dramatically elevates cancer risks for astronauts. But for a pharmaceutical researcher, this environment perfectly mimics cancer or aging-related lesions. It provides a natural, high-dose platform for testing radioprotective agents, antioxidants, and DNA repair enhancers. Plus, exposing microorganisms to this radiation acts as a natural accelerator for mutations. It helps us discover highly resilient microbes that produce completely novel metabolites, which, fingers crossed, could eventually help revitalize our stagnant antibiotic pipelines. To actually harness this rapid mutation, scientists are developing an advanced closed-loop system driven by artificial intelligence. It's a 3-step process. Step 1 is space mutagenesis, where deep space radiation forces biological adaptations. Step 2 is phenotype screening. Here, AI crunches through mountains of multiomics data, genomics, proteomics, metabolomics, the whole shebang, from these space-altered organisms. Finally, step 3 is drug target validation, where we rapidly identify the candidate molecules that show superior radiation resistance. AI acts as the ultimate pattern recognizer here. It can process years' worth of genetic adaptations in a matter of hours, parsing millions of data points to find that one golden molecule capable of surviving the extremes. Section 5, Formulations for Long Missions. Finding a new drug is really only half the battle. If we're actually sending a crew to Mars, we have to make sure those life-saving medications survive the journey with them. Standard terrestrial medications are surprisingly fragile. Without atmospheric regulation, spacecraft face brutal temperature swings, from a boiling 121 degrees Celsius down to near absolute zero. On top of that, high vacuum environments cause volatile drug components to literally evaporate right out of their packaging. And space radiation induces oxidation and hydrolysis, causing standard pills to lose their potency or even generate toxic byproducts. To overcome this, scientists are having to engineer incredibly advanced formulations. We're talking temperature-resistant liposomes, stable polymer nanocarriers, and specialized freeze-dried lyophilized formulations just to keep the medicine viable. Now, looking a bit further down the road, and I want to be super cautious here to separate established fact from future possibility, there is the highly speculative potential of using extraterrestrial resources. Conceptually, lunar regolith is rich in helium-3 isotopes, which could one day be harnessed for highly precise radiation therapies. Asteroids and meteorite fragments contain rare earth elements, like neodymium, that could serve as novel catalysts for drug synthesis or even enhance MRI contrast agents. So while mining asteroids for medicine is strictly in the realm of the distant future, the unique chemical building blocks found in space could fundamentally expand our understanding of biological chemistry. Which brings us to part 6, limitations and Earth translation. As amazing as all this sounds, we need to ground this explainer by looking at the harsh realities and very real bottlenecks of extraterrestrial research. So the crucial point is this. While deep space offers mind-blowing advantages for novel target discovery and high-quality crystallization, it is severely handicapped by its limitations. Space experiments are crippled by exorbitant costs, incredibly scarce launch windows, and a lack of unified quality standards. Down on Earth, we have brilliant reproducibility and mass scale, but we simply cannot perfectly replicate the combined effects of true microgravity and deep space radiation. It's a real catch-22. The good news? We are actively finding workarounds. The commercial space boom is drastically lowering orbital transport costs. At the same time, we're seeing the rise of miniaturized, automated microfluidic biochips. These are tiny, low-energy laboratories that can run complex fluid experiments completely autonomously in space, beaming data back to researchers in real time. Scientists are also relying heavily on ground space integrated workflows. Basically, they do the initial screening on Earth using magnetic levitation or advanced bioreactors to simulate microgravity, and then send only the absolute most promising candidates to orbit for final validation. And we really need these solutions because when we look at astronaut health holistically, spaceflight stressors don't happen in isolation. Radiation, microgravity, and severe psychological confinement all influence and amplify each other. This leads to incredibly complex issues like immunosuppression, neurodegenerative markers, and fluid shifts. Because these stressors are so interconnected, generic ground-based drugs often fail or trigger totally unpredictable side effects in space. We are essentially being forced to invent highly targeted, personalized medical solutions for these specific, extreme physiological states. And that leads us to the absolute core of why we do this. As the source research highlights, the ultimate goal is a bi-directional conversion mechanism. The objective isn't just treating a handful of astronauts on a Mars mission, it's about taking the high-resolution protein crystals, the radiation-resilient microbes, and the advanced drug delivery systems discovered in the extremes of space, and translating them back down to Earth to cure terrestrial diseases like Alzheimer's, osteoporosis, and cancer. So I'll leave you with this question to chew on. But forcing biology to adapt to the most hostile, unforgiving conditions imaginable, we are uncovering the hidden resilience of life itself. Will the void of deep space ultimately provide the precise blueprints we need to eradicate our most stubborn diseases here on Earth? It is an absolutely fascinating frontier, and honestly, the research is only just taking off. Thank you for joining me on this explainer, and keep questioning what's possible.